Hi, my name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I also serve as Chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Before we start the seminar, I just have a few bookkeeping items to go over. Questions will be responded to at the end of the presentation. Please enter your questions in the Zoom Q&A box, not the chat box, the Q&A box. The audience can prioritize the order of the questions by clicking on the thumbs up button. Closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. Instructions are also provided in the chat box. If you're having technical issues, you can make a comment in the Zoom chat box. With that, I would like to turn the proceedings over to our moderator, to our NCI Program Officer, Dr. David Miller, who is a Program Director at the National Cancer Institute's Division of Cancer Biology. Dr. Miller led NIH's Big Data to Knowledge BD2K initiatives, targeted software development program, supporting the development of data science software tools, and is active in NCI's Informatics Technology for Cancer Research, ITCR program, and Physical Sciences Oncology program. David is a computational physicist who maintains a grants portfolio encompassing mathematical modeling, data visualization, and interactive media development and research. Dr. Miller.
Thanks so much. Hopefully everyone can hear me. So thank you everyone today for joining the crossover seminar that we're having today that brings together 3 ongoing efforts under the banner of the Cancer Moonshot. So these are the Emerging Technologies Seminar Series, the DataViz plus Cancer series of events, as well as the Cancer Moonshot Seminar Series. So the Cancer Moonshot Seminar Series highlights exciting research being performed by investigators supported by the broader Cancer Moonshot Initiative. The Emerging Technology Seminar Series highlights the NCI's rich technology development portfolio and hopes to spur awareness and new collaborations as a means to accelerate the dissemination and adoption of these NCI-supported tools and technologies that lead on to benefits for cancer patients. And the DataViz plus Cancer series of events highlight opportunities for collaboration across cancer research and data visualization fields, including user experience design, game development, and immersive media. Across these, the next NCI event will be the final Microlab, featuring Sheila Carpendale, one of the pioneers of user experience, who hails from Simon Fraser University, in conversation with Cancer Moonshot awardee Dr. Lawrence Fong from UC San Francisco. You can register for this upcoming Microlab via the QR code here, as well as view recordings of all previous events. Now I have the great pleasure of introducing our moderator for today's seminar, Dr. Rachel Karchen from Johns Hopkins University. Professor Karchen is a computational biologist who develops algorithms and software to analyze genomic variation data, tumor evolution, and the adaptive immune system. She has a number of titles and appointments, as you can see here. However, I know her primarily for her development of OpenCravat, the widely used open source software toolkit for variant annotation, which has been supported for a number of years by the NCI's Informatics Technology Development Program, ITCR. Rachel, I'm happy to hand off to you.
Well, thank you for that introduction. Thanks everybody for coming. And I'm going to introduce our speaker, Zeynep Gamouche. Dr. Gamouche is an associate professor at the Department of Genetics and Genomics at the Precision Immunology Institute and the Tisch Cancer Center of the Icahn School of Medicine at Mount Sinai in New York City. She earned her PhD from Princeton University and pursued her postdoctoral research at University of Pennsylvania School of Medicine and Weill Cornell Medical College. At Sinai, she received the Faculty Council Award for Academic Excellence. She is also a recipient of the Outstanding Young Person in Academic Leadership Award by the Junior Council International in her native country, Turkey. Her research is at the interface of computational cancer genomics and data visualization, and she has worked extensively on simplifying the user experience to visually explore disparate, massive, complex data sets in major NIH-supported consortia. So Dr. Gamouche is going to speak, and please feel free to put your questions into the Q&A. And after the talk, we will go through the questions and discuss them. So welcome, Dr. Gamouche.
Thank you for this wonderful introduction. It's really a great honor to be here. My lab has been working at the interface of computational biology and data visualization for quite some time. And one of the questions that we are trying to answer is how to visually explore cancer omics data sets in the best way that's used for that in the best way for the user. And I want to thank the let me see. And I want to thank the NIH for putting together this and the Cancer Moonshot Initiative to put together this initiative because because it's very important in these efforts to be able to build a bridge between data visualization and cancer, and these kinds of efforts are working great towards this goal. And within the work that we have, we do computational oncology, web development, and visualization design, but that has to go hand in hand with investigators, clinical researchers in clinical oncology research, as well as basic researchers. And within this framework, my lab is in computational cancer genomics, and we try to identify and target drivers of cancer genesis and progression and therapy. And these are some of the topics that come from our manuscript abstracts, scientific study abstracts. And you can see that we are devoted to cancer research, but within that, an important component of our work is software and web interfaces for this purpose. And here are some of the tools that we have developed that I'll mention a little bit in more detail, as well as thanks to NIH-supported major consortia that generate data that we have been analyzing, in addition to other individual labs that we have been collaborating so far. And the first step in trying to explore a system is to make a picture of it. It makes life so much easier. For example, when we're looking at a cellular signaling system, the first thing that we do is to make a system blueprint. We may not know all the details of the system. We may have missing connections. It may be incomplete. But when we have a blueprint like this, even if it's a cartoon, it helps us understand the system better, but also to communicate the system better. And it's very, very important because what we do, for example, this is a signaling pathway inside the cell, and it's so far away from the reality. These proteins, they all have their own shapes. There's molecular crowding in the cell, etc. But if we want to just focus and look at the signaling pathway, it does the job, and it does a much, much better job than how we define these interactions mathematically. So this may be the correct or the more accurate way of describing the system mathematically, but in terms of communicating the information, it's much more complicated, and it's a little hard to make any conclusions or understand it. So, and it's important to be able to understand the system and rechange the system and work around with the system, because once we understand the system and communicate it, we can develop these mathematical abstractions and also simulate and even predict the system behavior for this pathway in the end to be able to understand the functional behavior. So cartoons have a role in even if it's a cartoon, visualization is very important. And even if we go outside of the cell, outside of the protein, and just look at social networks, it's the same thing. So this is an old slide that I enjoy putting up nevertheless, because this is actually a network of all my friends on Facebook, and how they interact with each other. So even if you don't know me at all, just by looking at the structure of the network or the interactions or the friends I have within and the particular patterns of interactions here, you can make a deduction about me or reach some conclusions about me. And the first thing is that I have friends. So at least in social life, social media. So you can say, oh, okay, she has friends and her friends are, friends with each other. But then if you look a little bit deeper, there's a little bit more to the story where there are little clusters, right? It seems like maybe in this cluster, even though I know everybody and I'm friends with everybody, they also seem to cluster on top of each other because they all know each other. So maybe I'm not that central in that network. Even if you take me out, there's still going to be a network there and interactions. And that's quite different than the majority of the bigger network, right? And what happened is actually these are all my friends from high school and college where I was in Istanbul, Turkey. And of course, over time, they all get to know each other. It's within a community of individuals. And however, some have been brave like me to come into the US and make friends within the US. And those are my connectors. They're in Turkey, they're also in the US, and they're kind of the connector people. But then, there are other clusters that I have, like institutional and etc. So just by looking at the picture of a network, and we can also make some conclusions. And proteins, coming back to the proteins or biomolecules, it's a similar situation. They can have disconnected networks, or they could have connected networks where we have connectors. So these could be critical. If I want to attack this network, I would target these guys so that it would break into two. And there could also be hubs. And there's a huge science of network science where different positions and the locations and centrality of the nodes and edge properties tell us something about the network that I'm not going to get into. But this is just to explain how network topology is important in our understanding of a system, even in a biological system. And people have been cognizant of this for a really long time. And there are general tools for developing networks, visualizing networks such as Gephi. And for biomolecular networks, there's Cytoscape, which is more focused on biomolecular networks, and it's getting both of these are getting updated regularly, and they're mostly in 2 dimensions. However, what happened was that when I started my postdoctoral work at Weill Cornell Medical School in New York City, we were actually fortunate to have a cave immersive visualization environment. And not every school had a cave, but the fact that we had it where I could wear 3D active stereo glasses and enter inside this immersive visualization environment. And here I am interacting with a brain, thinking that I'm inside of it, right? And here I am interacting with a network. This was fun and interesting. And what I wanted to do was also to visualize network within this cave environment. But the problem was that I had to use licensed software, and those softwares were really expensive. So I said, why don't we just, for networks, just for fun, why don't we just build our own network visualization tool? And that's how my foray into data visualization kind of started. And we developed this tool called IK, which is an immersive 3D network visualization tool. And here is a glioblastoma network, brain cancer network that is generated by the tumor cancer, TCGA tumor cancer genomics consortium. So when we look at this layout of the pathway that is critical in glioblastoma, we have a topology of the network, and it's static. It's in the paper, or even if it's on screen, you can't really do much with it in terms of looking at the topology in different ways. And here is Dr. Jian-san, a former postdoc in my lab, interacting with the network on his desktop, and here I am interacting with the same network inside a cave. And the thing is, when you have the 3rd dimension, what you can do is you can kind of move around the networks to get a snapshot that is easy for you to be able to understand the network better. And here, for example, with the right angle, with the right moving around, you can see that, oh, okay, so actually these 2 nodes may be critical because they seem to connect this part and this part of the network together. So if I'm going to target this glioblastoma network, maybe these could be good drug targets. So it helps you develop a hypothesis so that we can test or not test in the end. And when we go back here, these are actually part of a feedback control loop within this network, and they are critical, but it's not that obvious or intuitive when you look at it in the page versus you have a 3D rendering of it. And then we used IK for also visualizing networks with known 3D structure. You can look at the human brain, human neuron morphology, and even look at like a protein and bundle the edges to see like communication highways within a residue, which works well when you have communication highways, which is the case in this protein. But when you look at the human brain connectivity network, when we do that, because the brain is so plastic, there really isn't a highway of connection. That also, even when you bundle, you can make some deductions of the system through the visualizations. And then we were fortunate enough to participate in a study where we look at DNA variants that regulate the expression of genes. Here you're looking at about 9,300 edges, nodes, and 28,000 edges, and an animation of the network where basically DNA mutations or variants that are more central or bigger in this network are regulating more target genes. And that's the gist of this figure. And we published this in Science, and that generated more interest from the community to use our tools. So hopefully I can go to the next page. Okay. And then we could also look at like complex networks. So this is not in cancer, but it's a complex and dense network in schizophrenia that we published in Nature Neuroscience. And you know, we could look at multiple layers of networks, and depending on what the user wants, it can be visualized. So it was very powerful because we also, what we did was we implemented a lot of layout algorithms that we developed or we modified, which were super useful. And through that, for example, we could also cluster different clusters we could visualize, or we could do graph clustering methods to cluster it ourselves, and you know, different layouts, and we had this tool. However, there was an issue with iCave, and that was that it was a standalone tool. So you had to download it, you had to install it, but before you install it, then you have to have like the certain libraries, and with every iOS update, you have to make sure that it goes to the right iOS library location, which is kind of a mess. And even if everything works perfectly, users do not want to spend the time to explore networks by installing software, installing this and that. So it seemed like the evolution of this tool based on the user feedback was, so we started from the cave environment, and then we said, let's just go to the web. So now we're in the process of moving everything to the web, and we started this with the NCI sponsored Clinical Proteomic Tumor Analysis Consortium, which is CPTech. And there was an issue with CPTech where they generated a lot of networks, and I'm gonna skip the parts of how they were generated, and I want to focus on the visualization. And so they had they had these large dense networks, and then they needed a easy to access and easy to use tool with like they wanted to have the 3D capabilities because it's large and complex and dense, and there were also some customized analyses that needed to be done with CPTech. So we developed this. So this is like a snapshot of the network from the CPTech and the website, and this is the web address of it. And if we are to, let me see, to do a demo now of the tool. So okay, so this is the how CPTech tool looks like. And here we have the network, and it's not static, and the layout we were able to generate an iCave, and then we carried it to the web. And you can make some, you can look at the overall structure of the network, and here you can see that there are certain clusters of information on proteins and genes interacting. And then perhaps you can also look at a specific node. For example, here ERG, let's see, okay, ERG is right here, it's within the purple node, it's angiogenesis enriched, and then it's connected to these genes, and then you can see the grade, okay, so it's associated with clinical grade, so you can make some deductions and you can do some queries. And then you can even do this further and then look at this angiogenesis model in more detail, and then make other analyses with it, associated with it. So that was kind of that was kind of cool. And going back to CPTech, so that was our first start with network exploration. And however, the issue remains. So we had Cytoscape for general biomolecular networks, and we had that we could use for network visualizations as needed, which is a great tool, by the way. And then there's a CPTech interface that we had, but the CPTech interface was it did not allow us to give for the users to upload their data and visualize and interact with their data. So it was specific and dedicated to the CPTech networks. And there was a demand from our collaborators to make this in a way so that we can upload data to it, and then they actually they upload their own data so that we don't even have to be involved with the data visualization part, and they just do it themselves. And that we started working on this new tool called PhosNetVis, which is a phosphoproteomics network visualization tool. And the important thing actually now I want to talk in a little bit more detail is CPTech is for clinical proteomics data sets, and the data sets that we had was phosphoproteomics data sets. And what does that mean? Basically, the process of protein type called kinase phosphorylating or dephosphorylating its targets or substrate proteins is extremely important cellular signaling. And it's a little bit even more complicated because when this protein is phosphorylated, some of the residues some of the residues that it has can go up in phosphorylation, they can be neutral in expression, or the phosphorylation can decrease depending on what the process we're looking at is. Because once for example, a tumor occurs, perhaps the kinase activity is different, or when the cell is stimulated by a virus or any other means, or a therapy, then again, these phosphorylations change. But the most importantly is that when you have a phosphorylation or a proteomics experiment, all you get is the substrate protein and the phosphorylation site. You don't know who the kinase is. So the number one question is, who's the kinase? And for that, we kind of do an enrichment analysis looking at the kinase substrate interaction data sets where they already have information that is available on these. And so, for example, the most popular is Phosphocyte Plus kinase phosphosite substrate data set, and it has thousands of kinase substrate pairs and phosphocytes that we can look at. So to answer the question, who's the kinase? And we don't say who's the kinase, we say who's the most likely kinase, right, in the end of the day. And then there are more tools that are coming up in this area. But basically, first we want to be able to identify the kinase. And then once we identify the potential kinases, we also want to identify how the interaction network is changing. And this is different when it's a immune hot tumor. If it's responsive to immunotherapies, it's a hot tumor, and then it can have a different network, or it could be not that responsive to immunotherapy. So that makes it a cold tumor. And their kinase substrate protein interaction network could be different. The kinases involved could be different, or the phosphorylation levels could be different. So basically, we are looking at different networks of interactions. And so in FOSNetWiz, what we allowed is users can visualize their own networks, however they generated, like in the previous CPTech example, and then just use the tool. Or if they need a little bit more help, we can also we also added like a R-Shiny front end, so they can just upload their information, just their experimental data, differential phosphorylation data, and then we find the likely kinases for them through a gene set enrichment analysis, and then visualize the network. And here is like a network snippet. For example, here's the kinase, here's the target proteins, and here's a protein where we have multiple phosphosites, for example. And on the back end, we use Phosphosite Plus, and as I mentioned, FGSCA, R package, and the visualizations we did with 3GS and 3D Force Graph. And this is like a snippet of an input file. And voila, we get a network visualization. And if you're interested, you can go to FOSNetWiz.app and then do a visualization. And I'm going to attempt to do that on a live demo here. So this is the interface. And if we just wanted to go and explore existing networks, for example, here we have an example on SARS-CoV-2. And let's look at the actually pan-cancer data. And so this is another more recent CPTech immune pan-cancer data set that we're about to submit. And this is just a CD8 plus interferon gamma plus tumor types and the kinase activity there. And obviously there's a kinase that's highly active, and all its targets are also increased in phosphorylation. And if we want to know what it is, we just turn on the labels, and we see that it's PRKCA, and we can't see it clearly, but it's okay because, you know, it's interactive. We can just move it around and look at it in a different way, or, you know, zoom in and out, or just rotate it, however we like to do it. It's fine. We have complete control. And then we can also, if we are interested, we can change the background color to different colors, you know, and choose the colors we like. Maybe the green is better, and or just, you know, just go back to the original. And also we can take screenshots and etc. But we can also do like a node query, right? So we can also try to see what's going on a specific node. So for example, if I'm just interested in IRF3, then I will know what site and what is this association and what is the p-value, which means that what is the most likelihood that this guy is really phosphorylated, right? And where is it phosphorylated, and what are its values? So these are some of the ways we can use FOSNetWiz, and actually users are free to go ahead and use and apply this. So this is a study we're about to submit for publication, but we're always open to suggestions. And we're adding more examples. So with WebCave, within WebCave, I skipped the part, but we also participated in the PsycEncode consortium, and we did a web interactive network for a science paper as well. And but in terms of tools, we have a CPTech network exploration portal for NetView and FOSNetWiz that I just showed you. Feel free to play around with this, tell us what features you want to see, and we'll add them while you have time. And then we started working in another angle, we started developing this called immune regulation. So now we were committed to the web, and we decided to develop a web-based tool to identify human immune regulatory elements. We call this immune regulation. And because at the time, there were many diverse consortia generating massive amounts of gene expression regulation data, immune system changes, which could be vaccines, pathogen therapies, And we wanted to put all of these together and make them easily explorable. And while I'm presenting, feel free to go and interact with it in immunoregulation.mssm.edu. It's all free for all academic users. And here, what we wanted to look at was DNA variants that affect gene expression, which we call EQTLs. And why are these EQTLs important? And it's a funny name, I admit it. So if we have a mutation or a variant in an individual, and it may have no effect in the expression of a gene, or it can increase the expression of a gene, then it becomes an EQTL. This is not an EQTL. There is a mutation. Who cares? Gene is expressed no matter what. But some of the mutations are interesting, because even if you have a variant, nothing happens and it's okay. But only when there's a response, only when there's a stimuli like a vaccine, like an immunotherapy or disease, that this becomes important. This becomes a variant that impacts the expression of a gene, and that's called the response EQTL. And so these are the kind of EQTLs that we wanted to study more. And the other thing with EQTLs is that, just to make it more complicated in biology, they are tissue and cell type specific. In one cell type, they can affect this gene, and this variant in another cell type can affect this gene. And then they can be cis and trans, which means that they can affect the gene nearby, or they can affect the gene really further away. And in immune regulation, we wanted to put all of these together, the cis EQTLs or trans EQTLs or response QTLs. And we developed this largest central repository of data sets on gene expression regulation on whole blood and blood cell types. And what that means was we had about 220 million DNA variants based on data collected from 43,000 people and 50,000, I think more, experiments. And then we had specific data on whole blood, B cells, monocytes. And why is this important? All these blood and blood cell types, they're very important in the immune system. And that's why they were really important for us to look at. And also other infection data we had, like chikungunya, Zika, whole blood, etc. And when we were publishing it, there wasn't COVID at the time. And then we had, we also included transcription factor regulators of gene expression. So for that, we had about 736 transcription factors that led to 14 million gene region hits. And we said, why stop there? Let's also add all the DNA variants that impact a trait, like that increase your likelihood for disease or other traits. And we added 67,000 SNP trait associations. And here is the architecture of immune regulation. We have the data sources that we have, and then we process the data. And once we process the data, we make the data available. We used, for data visualization, we use D3, IGV, and then there are queries, functions, tutorials, and then most importantly, the user can query these, or they can upload their own private data. So I'm going to do a very fast, hopefully, demo on immune regulation. And I have one here. And so for example, if I am to search for JAK2, this is the main interface. And here we have any study. We added COVID afterwards. So you can see JAK2 in whole blood or monocytes or CD4 T cells, B cells. You can look at it anywhere you want. So let's see if I want to look at it in an LPS treated monocytes. So, and then this is the main interface that opens up. And here I can see that LPS in 2 hours in European individuals, because these DNA variants that impact gene expression, they're also impacted. They also change based on ancestry. And here, all the red variants are what we see in common in multiple studies. And the blue ones are just unique to the 24-hour LPS. And here it's 6 hours, actually, the response that disappeared, for example. And then we can also see which trait associated variants are corresponding to these. So we can also look at, and then what trait is that associated with, for example, and we can and this is like eosinophil counts, for example, for this disease that we can go in, and then here is like the location of the gene. And this is actually a fun tool because we can query for cis-eQTLs, trans-eQTLs, and transcription factors, and so on and so forth. So going back to the presentation, so immune regulation, even though we didn't really advertise it, and this is not most recent, it still had quite a bit of individuals that were interested in it. And these are all, we tracked all the visitors. And so far we had visitors from 43 countries, a few thousand individuals. And then we also tracked what they use. Most of them use the desktop, and this kind of information helps us feedback and develop the tool based on how the user accesses it and what they do with it. And then the user, usually they do direct, and some of them learned it from social media or referrals, so feel free to tweet about it. And so that's the immune regulation tool in a nutshell, which was the largest database of immune regulation data. And it's an interactive interface to visualize and discover association between genes and their regulators, and by regulators, we look at transcription factors or DNA variants of the individuals that are associated in specific studies and associated with immune research. And how is this useful? Because it helps us explain cohort or cell type dependent variations, select the cohorts, the right cohort for follow-up studies, and develop molecular signatures or immune response or use in future applications. And here is the, and we had a publication from it in NAR. And from the remaining time, I want to talk to you about a project that we are working on. We just recently started working on, and that's called PRIMAVA. And that's within the context of a Cancer Moonshot grant that my group has received. And PRIMAVA stands for Precision Immunology Monitoring, Analysis, and Visualization Online. And that's a handful. So what does that mean? One of the priorities and important issues within the Cancer Moonshot Initiative is to be able to understand who will benefit the most from cancer immunotherapy. So if we have a population of cancer patients such as here, and let's say the ones in red are excellent responders and the blues are not going to respond well, why should we make people suffer through going through these treatments, right? So we want to be able to precisely target the right treatment for the right patient in the right concentration and the right dose and the time period and combination. So in order to do that, the Cancer Moonshot Initiative has put together the network of researchers from the Cancer Immune Monitoring and Analysis Centers, which is CMAX, and we have one at Sinai, and the PI of that is my collaborator, Sasha Ginyatic, and also the Cancer Immunologic Data Commons, CIDC, where a lot of the data analysis takes place. And why is this needed? Why do we why do we need to have a large consortium and large initiative of investigators? And the reason is, when we look at cancer immunotherapies, it's most of the time it's not a single predictive marker that we're going to look at. It's not going to be one assay. It's going to be a comprehensive test results, and based on this comprehensive set of test results, we need to make some predictions on who's going to be able to respond ideally. And for this, actually, there are disparate immune monitoring omics data sets. And here's just, it's not a complete list, but for example, protein analyte expression, gene expression, cell abundance, antibody levels, genetic mutations as needed, microbiome analysis as needed, and in situ characterization of immune cell infiltrates, and also newer and more expensive technologies such as spatial sequencing or single cell sequencing, which generate massive amounts of data and are expensive technologies, these last two. But however, all of these are different technologies, different analysis, harmonization steps, and what do you do with that afterwards? How do you put it all together? And once you put it together, how do you share it and make it available to the community so that you can advance and accelerate the speed of discoveries associated with cancer immunotherapies? And not not to make the problem even more complicated, all of these analytes are also measured at multiple time points. So you do for the patient, first you look in immunotherapy trial, you look at the baseline immunocompetence, and then during treatment you look at the dynamic changes in the treatment, and then after treatment you look at the immune correlates. So all of these have also multiple time points, and with the objective of trying to look at this. But why do we do all of this? Because it makes a difference. So here's an example of a immunotherapy clinical trial response. And where here we see, for example, in treatment 1, we have complete recovery and partial recovery, but also progressive disease, for example. But in treatment 2, we don't have progressive disease in the patients. But if you look at treatment 3, we have complete recovery, and then none of the patients have progressive disease, which is fantastic. And you see how the number of patients has increased in complete recovery. So this makes it all worthwhile, worth all the effort to put into this. And so Primavo is a web-based tool for visual analysis and interactive exploration of immune monitoring assay data sets from Cancer Moonshot Initiative. And from CIDC, we have all this different assay type data sets, but we still, if they are harmonized, we take them, or we will take them from CIDC, but we also have analysis in Sasha's lab and in Minerva, the supercomputer of Mount Sinai. And then within our data repository, we have image data, dimensional reduction, gene expression. We can do correlation and network visualizations. And for the visualization, we used multiple different tools. And the objective was to be, so our objective, and this is a project still that we recently started and still under development, is to have like a query browser, cohort, and assay browser, and so that we can, and most importantly, a user feedbacks, because at every step of its development, we are working with individual researchers and investigators, and feel free to reach out to us if you have any suggestions or if you want to work with us. We love interactions and teamwork, and the user basically can upload their data, share or download their data. And front end, we wrote it in TypeScript, and backend is in Python. And I'm gonna, before I conclude, I'm gonna do a really quick demo on, hopefully, on Primavo. And this is the study selection page of Primavo, where you can choose different clinical trials, and you have like now how many samples and how many patients are there. And you can also go to clinicaltrials.gov to know more detail. And just note that the study is currently, because it's under development, we have a password protection, but that's going to change in the upcoming months. And once we go to the data selection page, here is what it opens up. So here we have, for example, a study with 3 different therapies, and with 3 arms in each therapy. And here you can see different drugs and how many patients there are in that study. And then we have an escalation where the drug dose is increased, and here we have an expansion, we have more patients there. And then we have 19 patients in treatment 19 patients in treatment 1 and 16 in treatment 2. And we can do all these, we can select any of these, and then interactively we can go and look at the data, which I will show you in a minute. And then for this specific clinical trial, we have serology, Olink, and CyTOF, and I selected Olink and CyTOF. And then we can see what cancer type the patients have, which is Hodgkin's lymphoma, and the race distribution of the individuals. And then, we can, if we only want to look at the white patients, we can do that immediately. And for some, and then the numbers change automatically. So now we see Olink is this many, CyTOF is this many, and this many cancers, their distribution. We are populating this selection area, so we're going to have more information. We also have the gender, we have the time point, how many patients have which time point, and also their responses, like response, partial, complete response, partial response, and so on and so forth. And then this opens up the data exploration page. And so in the data exploration page, what we can do is so what we have is just summary. And because we just chose Olink and CyTOF, we're only going to have the patients we're interested in, gender or ethnic ancestry, or we're going to add age group, whatever patient we're interested in. And then we can look at it in more detail. And let's see, if I look at Olink data, which is protein expression data, and just click on CXCL13, for example, which is a marker of inflammation, and CXCL13 is usually worse in non-responders. And as you see, as the treatment as this treatment moves on, the CXCL13 levels decrease with the treatments, which is a really, really good thing. And you can see which patients have the highest, which is not that great, and which patient has the lowest CXCL13, which is good, just by looking at this study. And then maybe we want to look at this a little bit more in detail. And then if we compare, for example, complete responders and partial responders here, and what are the levels of other protein markers. And we here see that, for example, complete responder and partial responder, this gene is good. And then here they are low. And here you see that CXCL13 is actually low in this set, because we want, in complete responders, it's good that it's low, and it's a marker. And we can do a similar study for CyTOF, and CyTOF measures the specific cell types that are associated with the system. So here we can perhaps look at dendritic cells and the levels of dendritic cells. And here you see that it's interesting, because the dendritic cells are increasing with time over treatment. So you see it increases with treatment, and then off study, and then it goes down and it increases again. And then we can also look at maybe partial response. Let's see, complete response versus like here. And then we can see that partial response and disease and progressive disease. So these are good guys. You want these markers to increase. So that means that things are working, and you want these ones down, because they are signifiers that things are not going that well. So so that's it in a nutshell in terms of where we are with our studies. And we are very excited to develop Primavo, and because we believe that once it becomes active, it's going to be a huge resource for the immunotherapy community, immune community, because they can filter, query, visualize, share myriad high-dimensional data sets, and towards that we also care about our user-centered design principles. So we want your feedback and input to make it better. And here's the acknowledgement slides, and I'm on sign, and we have multiple collaborators and different consortiums, but most importantly, Kevin put huge hours on the development of Primavo, and Bark put huge hours again in FOSNet-Viz, and all the data we got from Sasha and Edgar, we couldn't do it without them in terms of Primavo. And alumni Selim, who is a faculty right now, he's a newly minted faculty. And so he worked on immune regulation and other earlier, and ICAVE. And with that, we love feedback, and this is my email if you'd like to email me, and I'd love to answer any questions you may have. So.
Well, thank you, Dr. Gamouche. That was wonderful. I see right now we have one question in the Q&A. I hope there'll be a few more. But the first question is, the system seems generally applicable. Is there any work for the glycogene network?
Yeah, absolutely. You want to visualize glycome network? Yeah, you can do that. If there are any specific issues unique to that network type, we'll have to see if the tool accommodates, but if it doesn't, let us know. So maybe we can collaborate together.
All right, the next question For Primavo, could you describe the patient populations that were used for the input data? For example, which CTs were used?
Mhm.
And also, how diverse are these patient populations?
Yeah. In terms of the data generation, we don't have any control. So these are immunotherapy clinical trials. So it's the individuals who come to the clinic and who consent to be a part of these studies. And we do have a ancestry distribution. And in the example I showed to you, it was about the majority was again European ancestry, but these are a little bit earlier collection of data sets. And I think the clinicians who are generating data are cognizant of the fact that perhaps these clinical trials are more accessible in more affluent areas, which have more concentration of individuals of European ancestry. But there are efforts to be able to address these structural systemic inequalities, but we use any data that we can get. We are data agnostic, and we are very excited to be able to use different data sets. Yeah.
All right, the next question. For iCave and CPTech Viz, what biological metrics can be quantitated using the width of the connecting lines?
So that's a great question, because every now and then we get this question. So in a network where the 3D, where it has a 3D biological meaning, the edge length also has a meaning, right? And so, for example, in the brain, you can't put things together because they're far apart. Or a protein, like if the residue interaction network of a protein, you can't change, and that has a biological meaning. So in those situations, it has a biological meaning in the sense that, yeah, residues are far apart, the brain regions are far apart. However, if we are visualizing abstract data, the way we have layout algorithms right now is that we do not have the edge length having a physical meaning. We can associate a physical meaning to it. Is that we can add a layout algorithm where they are all, like, based on their association, they're more huddled together. But we don't have such a layout algorithm right now. But it's one of the things in our to-do list.
All right, here's a comment. Really cool stuff. Have you thought of involving graphic designers or interaction designers into maximizing the clarity and flow of visual representations?
Yeah, that is such an excellent question. And that's the question we have been debating within our group, because I think interacting with visualization design expert and graphic design experts is a wonderful suggestion. And I think that's what's going to happen moving forward. And I do have students from a master's program at Cornell Tech that a couple of them do visualization design. And for their project, we get help from them. And we've had the students for a few consecutive years now. And what I have observed in my own personal experience is that it's a lot. There's a lot of biology, and the person needs to understand the biology in order to be able to help us with the design and understand the biology and understand the user. So it is a full-time committed effort. And I think if there will be opportunities through NCI to fund such individuals, I think that would be wonderful. And then we could work with more visualization design experts. But moving forward, that's one of the things in our mind. And it's always a balance of where do we want to invest most of our time and efforts and funding for. But it's an excellent point. And I think the more data we have, we may go through that route. Great.
All right, here's the next question. Can you speak a bit more about large immersive data visualization like the CAVE or VR? What tools or approaches help you get to understanding the data rather than being overwhelmed by the data?
Yeah, so I think it's important what kind of data you're interacting with. So in our experience, for example, if you're looking at layers of information, for example, if it's a brain and you can look at it in different layers, and like and different angles. And for example, if you can zoom in from the brain to like a lower, for example, even like the interaction of proteins within the brain or the tissue layers, etc., or the partitions, then it's very useful. With networks, it was, as I mentioned, I just started as a fun project, and then it just evolved into something. And I think in terms of networks, it is probably more useful when you again look at a system that has a biological structure in 3D, like the brain, and you have the network and brain. But for the by and large, the feedback we had was people wanted something simpler. But this is back then. This is a while ago. And now where are we? People are looking. People are doing immersive 3D and, for example, point cloud visualization with UMAP, or and or even PCA stuff, and more importantly, and with protein structure work, there's a lot of immersive visualization. But most importantly, and I think the critical question is what's going to happen with metaverse? Because if our people are, because they've been time and again efforts of doing movies in immersive 3D and etc., and then there's a pushback, right? But it seems like now the technologies are maturing and people are getting familiar, because what I cannot do is I cannot make an experimentalist or a clinician get familiar with 3D immersive visualization. But if they come in, they've already done it in metaverse because of whatever reasons, for a lot of other things, and it becomes a thing, then it's going to be much more accepted by the scientific community and the visualizations we do once that kicks in. But we're going to have to see if it's going to kick in or not. So until then, we're more wedded to the web platform.
Okay, thank you. In addition to the 2D and 3D visualizations, is there a way to add time as a fourth parameter?
Yeah, absolutely. Actually, I didn't show that. I think I forgot to show that. For example, oh yeah, I didn't show that. So for example, with COVID, we have a COVID network that Bark visualized, and for the FlowSNIT Viz tool, what we do is we can have a simple animation. So basically you see how the network changes over time, and we had, I think, 4 or 5 measurement points, and then you can see which sites are phosphorylated, then they increase in phosphorylation, and then they decrease or they change completely, right? So, and it's really cool to be able to see that as an animation. And that's when the 3D becomes really beautiful, because while it's animating, you can rotate and interact with it too. And that gives you the time dimension.
Okay, here's the next question. Do you face pushback from users on adoption of more advanced visualizations? For example, SAMKEYS and networks.
Less and less. So I think as I showed on the bridge, so we're kind of forming the bridge, and I thought of adding this code to my presentation, and then I removed it. But basically, when I first started as a postdoc at Cornell, just completely computational person in a medical university, I met with a clinician scientist, and his question to me was, in our first meeting, so what can your computers do for me, right? So, but that was in the, a long time ago, and you can tell that this was in the nascent time period of big data, even like computational genomics was just starting and blossoming. So, you always have pushback, and I think we will also have pushback in data visualization, and we do. I mean, even a few years ago, I would say, oh yeah, I'm doing this visualization, and people would be like, why? Who cares? And they absolutely did not care how the design looks. But now look where we have come. The fact that someone in the box asks like, how about graphical designers? How about visualization designers? 5 years ago, if you asked this to a scientist, they would be like, no way. and you would be like, wait, actually this is useful, and it would take a pushback. So things are evolving, people are getting familiar, very comfortable with the web technologies, and they want to see things that are pretty. If in your daily life you see things pretty, why do you want to have or easy to understand, let me put it that way, and why do you want in your scientific life to look at like clunky interface who sometimes works, sometimes doesn't work? You don't want that. You want something to be easy to understand and clean, and unfortunately, even the most ardent opponents, when they see things in 3D, they love it, but to get there can be a pushback. So for anyone who wants to do data viz, I would just say, just do it and then tell them, or or just don't give up, because when you say I'm going to do this, I'm going to do that, people are like, yeah, I don't know. Yeah, and that always happens. You have to prove it.
So Rachel, we're at the top of the hour.
Okay.
Yep.
All right. Thank you, everybody.
Thank you.
So again, I'd like to thank our NCI program officer, David Miller, and certainly Dr. Karchin for an excellent moderation job, and our speaker, Dr. Gamouche. Please join us December 15th for our next Moonshot Seminar Series, Molecular Dissection of Tumor Biology, presented by Dr. Rabola, Dr. Suzuki. And with that, thank you again for an excellent Moonshot Seminar.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4-CANCER.